Hi everyone. In this video we will be seeing regarding myoglobin, their structure and their functions. What is the physiology of hemoglobin and myoglobin? That is how oxygen is binding to hemoglobin, under what conditions they are binding. Then the transportation of carbon dioxide, that is in what form carbon dioxide is transported to the lungs. And various poisoning effects of other ligands other than oxygen molecules that are binding to this ion. First we will see about myoglobin and their structure. Maybe you might have learned about hemoglobin. Hemoglobin contains 4 subunits. Myoglobin is nothing but very similar to hemoglobin, but it is like one subunit of hemoglobin. See, you can see here the structure has 2 parts, that is the heme part and the globin protein part. So I hope you know what is heme group. So this is the heme group. It contains iron as the metal, that is, it is a metalloporphyrin. So this is the porphyrin structure and it is quadplanar with around 8 substituents present here. So this is the heme group. This is also called Fe protoporphyrin 9. So here only one heme group is present, and this is the globin protein that is made up of various amino acids, like around 153 amino acids. What is the function of this myoglobin is, they will store oxygen. Why do we need oxygen? In order to produce energy, we need oxygen, that is to undergo cellular respiration, to breathe in and breathe out, we need oxygen. There are 2 main things, deoxymyoglobin and oxymyoglobin. In the case of deoxymyoglobin, the oxygen residue is not present, but in the case of oxymyoglobin, the oxygen residue is present. So this is the structure of deoxymyoglobin. So this is the heme part and this will be surrounded by the protein part. So the protein part is not mentioned here. So what is this group? This is the histidine group. So histidine, this is the histidine structure. It contains the imidazole part. So this is the imidazole part that is present in histidine group, and the nitrogen present in the imidazole is attached to our Fe. So what is the structure of this heme group? They are not planar. It has something like a pyramidal structure and it is also a square. So it is having a square pyramidal arrangement, but they are not planar. And as you can see, the Fe is in its +2 oxidation state. So if the Fe is having +2 oxidation state, then it might be having d6 electrons, because Fe is having argon 3d6-4s2, so that Fe2 plus will be having argon 3d6. The 2 electrons are donated. So these are the various energy levels where these 6 electrons are arranged, and as you can see, they are not paired, all are in high spin. See, 6 electrons, so 1, 2, 3, 4, 5 and 6. They are not paired. If they are in low spin state, they might be paired, but here this is in high spin. So these are all very important questions that are asked in various exams, that is in what state, that is in 2+ state, or they are in 3+ state, or whether they are in high spin or whether they are in low spin, and their structure, geometry, everything. So there is one electron that is present in this dx² minus y². So this electron present here, and the electrons that are present in the heme group, that is in the nitrogen atom of the heme group, and the electron present in the dx² minus y² level in Fe, there is electron-electron repulsion between them. So due to this electron-electron repulsion, the Fe2 plus is a little bit larger in size, so that they won't fit into this hole that is provided by this porphyrin structure. So in order that they will be little bit above the ring, that is out of the plane, 0.42 angstrom out of the plane. So they have a penta-coordinate iron 2 center. So this histidine group is in the proximal site. So anything that is attached to your downside is called the proximal site, and if something is attached above, then it is called the distal site. So coming to oxymyoglobin, so here nothing is the difference, but oxygen is attached, and as you can see, oxygen is attached in the distal site. So oxygen is not in the O2 form, but it is in the O2 minus form, that is superoxide form. And as you can see, Fe is not in the +2 state, but they are in the Fe3 plus state. So how is this bond formed means, one electron from the oxygen and one electron that is donated by the Fe, that's why it is converted into Fe3 plus, they will make this bond. And totally O2 minus has 13 electrons, that is 1 electron extra than O2, so they are superoxide. So see how the electrons are arranged in various energy levels. They are in low spin and they are paired. So since it is Fe3 plus, the total number of electrons is D5, that is orgon 3D5. So 1, 2, 3, 4 and 5. They are in low spin. Then see the structure Fe, O and O. They are like bent, right? So it has a bent structure and the angle here is 115 degree. Then you might have a question, how is this O minus stabilized? There are many amino acids in the protein, that is around 153 amino acids. Of course, there will be some histidine amino acid that is present in the distal site. So that histidine amino acid, this hydrogen group, the imidazole hydrogen group, and that O will make a hydrogen bond, as I have mentioned here, so that this superoxide is stable. And then this is having an octahedral arrangement, since there are around 6 bonds. So coming to the physiology of hemoglobin and myoglobin, before going into that, we need to know about what is partial pressure. Consider these two beakers. Here you can see that 0.60 moles of hydrogen is present, and here little bit more, that is 1.50 moles of helium is present, and here the partial pressure is little bit lower than the helium. So this is, partial pressure is nothing but the pressure that is exerted by a particular gas, either a hydrogen or a helium gas, at a particular volume. As you can see, both of them having the same volume. So partial pressure is the pressure exerted by a gas at a particular volume. So that is called partial pressure, and using this partial pressure we can find the total pressure using Dalton's law. So if we add up these, it will give the total pressure. And there is one thing you have to notice, that pressure is almost directly proportional to the concentration of the gas present there. As you can see, the concentration is low and the pressure is low, and the concentration is high, so the pressure is also high. So physiology of hemoglobin and myoglobin. So this is the diagram you have to see. As you can see, the partial pressure of oxygen in the lungs is like around 100, it is very high. So at this partial pressure, when the partial pressure of oxygen is very high, as you can see, the saturation of oxygen, percentage saturation of oxygen is high for hemoglobin, and myoglobin is not even attached at that stage in lungs. Hemoglobin is having around 96% saturation of oxygen. What we can find is, at high partial pressure of oxygen, hemoglobin has high affinity towards oxygen. And as you can see here, when the partial pressure of oxygen is low, that is in the case of muscle, hemoglobin saturation of oxygen is only around 50, but myoglobin saturation is around 90, I guess, 90. So this is because at this stage myoglobin has high affinity towards oxygen. So these are the two equations. In lungs, one hemoglobin can bind 4 oxygen molecules and it will become like this. And again this will bind with 4 myoglobin because one myoglobin can accept only one oxygen molecule and it will give like this. So this is occurring in muscle tissues. Another factor is pH. So I hope you know about cooperative effect. So cooperative effect is found in hemoglobin, where once the oxygen is binding to one of the subunits of hemoglobin, the salt bridges between the 4 subunits are broken. So that once 2 oxygen molecules are bound in the 2 subunits of hemoglobin, initially they are in the tense state unless they are binding with the first 2 subunits. Later on, after binding with 2 subunits, that is after binding oxygen with 2 subunits, it will move into a state called relaxed state, so that the other 2 subunits will be easily binding to the oxygen. So the overall effect is called cooperative effect. So this cooperative effect is pH dependent. That is, if the pH is high, then the cooperative effect is high. But if the pH is low, then the hemoglobin affinity towards the oxygen is also low. So this was given by Bohr's effect. That's why it is called Bohr's effect. So in muscles, more of the CO2 is released because glucose is converted into energy in the muscles, right? So more of the CO2 is released, so that this CO2 will cause an acidic environment. As you know, acidic means 1 to 7, so the pH is low. So since the pH is low there, hemoglobin will not have affinity towards oxygen. Instead, our myoglobin will have affinity towards oxygen. And in this muscle, the partial pressure of carbon dioxide is high. Since there is a more concentration of carbon dioxide in muscle, the partial pressure of carbon dioxide is also high. Coming to the transportation of carbon dioxide to lungs. Carbon dioxide is not transported as it is. It is transported in their bicarbonate form. As you can see here, this is the bicarbonate form. That is, majority of them are transported in the bicarbonate form, that is HCO3 minus form, and some of them are transported in the carbaminohemoglobin form. First, we will see the mechanism of this bicarbonate form. So initially carbon dioxide is present in the muscle since there is more concentration of carbon dioxide. So always we know that something will move from higher concentration and it will try to move to the lower concentration to attain equilibrium. That's why now carbon dioxide is now entering into the erythrocytes. In this erythrocytes, the carbonic anhydrase enzyme is there, and this carbonic anhydrase enzyme will convert carbon dioxide and water into carbonic acid. Carbonic acid is nothing but H2CO3. After carbonic acid is formed, it is immediately dissociating into H plus and bicarbonate ion. How this dissociation is facilitated? This is because of the deoxyhemoglobin. What this deoxyhemoglobin will do is, this is having some protein part in it, right? That protein part will accept the H plus ions from this H2CO3, so that this HCO3 minus is now remaining. This bicarbonate ion now will travel via the venous blood and it will reach the lungs. Now this bicarbonate ion has reached the lungs. In the lungs, we know that deoxyhemoglobin will be present and it will be binding with oxygen. And while it is binding oxygen, it will release that H plus ion that is bound in their protein, that was bound earlier in this stage. Now this H plus ion that was released from deoxyhemoglobin will again bind with this bicarbonate ion and it will give carbonic acid. So now carbonic acid is formed. As you can see, the transportation is fully covered by the bicarbonate ion. Once it has reached the lungs, it is formed into carbonic acid. There is this carbonic anhydrase enzyme which will again convert this carbonic acid into carbon dioxide and water. Now this carbon dioxide is exhaled via the alveoli that is present in the lungs. Around like 10 to 15% of carbon dioxide is transferred in the carbaminohemoglobin form. That is, initially the carbon dioxide is binding with the hemoglobin molecule, that is hemoglobin protein part, that is NH2 part, and it will be becoming carbaminohemoglobin, and H plus is released. There is no enzyme involved in this process, and later in the lungs it will be released as carbon dioxide and the hemoglobin will be present as it is. So poisoning effect of carbon monoxide and other ligands. So why carbon monoxide I have mentioned specifically means carbon monoxide will bind like 200 times stronger than oxygen with the ion. Why is it? Because it is a very strong ligand and it is a soft ligand. We will see about it. So here there are various categories of metals based on their hard nature, borderline nature, soft nature. You might have learned in organic chemistry regarding HSAB principle, where hard will react more with hard and soft will react more with soft. Similar in this case, metals and ligands are separated in their various forms, and soft metals will react with soft ligands more and hard metals will react with hard metals more. And the borderline, someone which is in borderline may react with soft well or may react with hard well, we can't predict. So here Fe2 plus is in the borderline, but it will react with soft ligand very well or strong ligand. But our oxygen molecule is neither a soft one or nor a strong one, but it is a quite good pi acceptor. That is, we need is, since Fe2 plus is donating electron, so oxygen has to be a good pi acceptor. So because of that it is binding. But ligands like carbon monoxide, cyanide and PF3 and more are very good acceptors, and some of them are even soft ligands. As you can see, CO is a soft ligand, cyanide is a soft ligand. So they will be interacting very well than our oxygen molecule. So see, as you can see, carbon monoxide even shows similar cooperative and Bohr effect like oxygen. So that what happens is the oxygen is not transported to our body, they are blocked, and it will lead to asphyxia. That is, there is no enough oxygen for the patient to breathe. So that's it about poisoning effect of carbon monoxide and various other ligands. So my references are Principles of Bioinorganic Chemistry by Lippert and Burke, and Organometallic and Bioinorganic Chemistry by Ajay Kumar. Hope you like this video. If you have any doubts, of course you can mention in comments. I'll reply. Thank you for watching this video. Thank you for supporting me. Thank you.